Exercise could activate the pleasure circuit and, consequently, it could be an addiction. Exercise has an antidepressive effect that reduces changes in our brain, increasing brain-derived neurotrophic factor. It has certain benefits, such as increased pain threshold or reduction of acute anxiety. Runner's high is an after hard effort period of playfulness and relaxation that increase endorphins and enkephalins that are endogenous opioids. These endorphins, beta endorphins, doesn't cross successfully blood-brain barrier. To measure runner's high, we use a brain scanner. We test 10 amateur distance runners while they were running for 2 hours, and they show an increase in opioids level in the prefrontal cortex, anterior cingulate cortex, and insula. Exercise also increased levels of endocannabinoids that can cross successfully the blood-brain barrier. Endocannabinoids and opioids can indirectly activate dopamine cells of the VTA and thereby stimulate the medial forebrain pleasure circuit. Pleasure and pain can be felt simultaneously. As an example, long-distance runners. Both indicate salience, and emotion is the currency of salience. It was proved that orgasm can be dissociated in sensory discriminative and pleasure emotional components that are mediated by separated brain regions. The same occurs with pain. There is a sensory discriminative pathway involved in touch and muscular sensation called the primary somatosensory cortex, a parallel pathway involved in the emotional sense of pain that contacts two emotion centers, the insula and the anterior cingulate cortex. People who sustain damage limited to the lateral pathway will report an unpleasant emotion reaction to a painful stimulus, but will be unable to describe its specific qualities or to locate the painful region on their body. Selective damage to the lateral emotional pathways results in the opposite condition, called pain asymbolia, in which people can report the quality and location of a painful stimulus, but not the emotion. The terms emotional pain and painful social situations. Is this just only a metaphorical language, or do we actually experience certain powerful emotions the way we do physical pain? Evidence indicates that emotional pain activates the medial but not the lateral portion of the physical pain pathway, and experiments of social pain have demonstrated significant activation of the insula and the anterior cingulate cortex. So, emotional pain is not just a metaphor. It partially overlaps with physical pain. In an experiment related to dopamine release from VTA neurons, it wants to produce a painful stimulus injecting a concentrated salt solution into the jaw muscle during an hour. The main result was that this long-term painful stimulus was associated with increased dopamine release in both the dorsal striatum and the nucleus accumbens. In the latest was seen the greatest dopamine release by those ones that reported highest emotional pain ratings. In order to interpret these findings, it was done an experiment of brief painful food shock in rats, and it showed us that there were two different patterns of response to the painful stimulus. First, dopamine neurons in the dorsal portion of the VTA were transiently inhibited by food shock. Their firing rate decreased below background levels. This one is the classic pleasure circuit, which is activated by pleasure rewards and inhibited by pain. In the ventral portion of the VTA, dopamine neurons were transiently activated by the food shock. It is a salience circuit that is activated by both pleasurable and painful stimuli, and it tightly coupled to emotional responses. To sum up, we can say that all brief painful stimuli eventually end, and the relief from pain we experience as they do end is itself pleasurable. However, chronic long-lasting pain is likely to produce long-term changes in the brain's pleasure circuit through the action of stress hormones. It creates a supersalient response in the medial forebrain, and that this somehow contributes to the popularity in some quarters of sadomasochistic sex or even of tasty but really spicy food.
Another virtuous pleasure is meditation, which must be self-induced, must involve progressive muscular relaxation, and involve a reduction in logical processing. But as Richard Davidson said, meditation is a whole family of practices, like sports, as each one focused on a specific point. It's incredible the work that the brain performs in a state of meditation. By brain scanners, the activity was associated with dorsolateral prefrontal cortex, anterior cingulate cortex, hippocampus, and striatum, possibly including the nucleus accumbens. Furthermore, practice of meditation over many years can also influence the patterns of brain activation. But does meditation activate the medial forebrain pleasure? Hans Lowe performed brain scans while people was listening to speech with eyes closed as a control condition and found an increase in dopamine release, what means that meditation and pleasure are correlated. Another interesting question is if mystical or spiritual experiences activate medial forebrain pleasure circuit. Biergaard explored this question performing by brain scanning on a group of nuns who spend most of the day praying. The ultimate goal is achieve unio mystica, a state where one feels completely united with God, but that's too complicated, so the brain scan was useless. Biergaard suggested instruct nuns to remember their most intense mystical, but that didn't make sense. Neither memory produced the patterns of brain activation that are seen in actual scans of subjects who has achieved the unio mystica, even though he lied later saying yes. The fact of making a charitable donation also produced an activation of the nucleus accumbens, a neural substrate of pleasure reward, and some theories hold this information. The first say that people give to charity out pure altruism, and the second one, called warm glow, holds that people like drive this pleasure, what means that they like to make their own decisions. We can see that in a Harvard experiment, where each subject received an amount of money and scientists proposed them to earn more or donate the money to charity. And the charitable giving produced more activity in the pleasure center than received money. As we know, this result doesn't reflect condition in the real world because the context has an important function. For example, a positive context also can activate pleasure centers. Also, as we said before, receiving money activates pleasure centers. But sometimes comparison with other people influences as a factor in driving economic decisions of individuals. We seem to be hardwired to compare our own experiences and circumstances to those around us.
We humans usually anticipate events that have not yet happened. We want information about what is going to occur in the future. But what does this have to do with pleasure? Is it pleasurable for us? Does it activate BTA dopamine neurons as food and water do? To answer these questions, an experiment of two thirsty monkeys was done. It consisted on two targets that were presented in a video, and those monkeys had to choose one target. Both targets had an amount of water as a reward for monkeys, so it doesn't matter the target they choose. But one target showed meaningful information about the reward that they were going to receive, that we can identify as information about the future, and the another one without any meaning. And finally, although monkeys were able to receive a reward in spite of the target they chose, they preferred to choose the target who had a meaningful information about water than the one who had no sense. Also, this experiment showed that neurons in monkeys' BTA increased their firing rate when seeing the target which predicted a large amount of water than the one which predicted a smaller one, what means that those neurons increased when the information from the target was meaningful. So we can say that the merely fact of knowing makes pleasure increase. Another issue that seems to make pleasure increase are abstract mental constructs, also known as abstract ideas, which are called as a superpower. That means that you can act according to your ideas and not to your basic needs, as happens, for example, with religion and sex. But how does this superpower, in which ideas engage the pleasure circuit, develop on a cellular level? The answer is not known yet, but it is true that experience can change the electrical signaling function of neurons, and what is more, it can increase or decrease the strength of the synaptic communication, as happens with LTP and LTD. And in regards to the brain part, these changes occur throughout the brain and take place in neurons of the medial forebrain pleasure circuit. All this comes because pleasure and abstract ideas seem to be related. But how can it be possible? How can pleasure be transformed in abstract ideas? Well, that's the role of the associative learning. Associative learning means that we learn by the association of different events, in this case, pleasure and abstract ideas, and that we need to use the long-term memory to be able to store information from both events to remember its association. According to the model, the transformation of pleasure in abstract ideas can be done by a fast associative learning, what means a reward prediction, a medium one, which means given ideas as a reward, and a slow associative learning, which can produce an addiction due to the fact that it can change the function of the pleasure circuit. So, to sum up, the experience ability to make changes in pleasure circuit has enabled abstract ideas to feel pleasurable, but unfortunately, this process makes pleasure to be transformed in addiction.